using in-hand sampling for KARAS. But before we start the season, I think I would once again like to invite one of the co-coordinators of this program, Dr. Sintu Kumar Samanta, to kindly deliver an insightful introduction of our honorable speakers. I would now like to hand it over to Dr. Samanta sir.
Okay, thank you, Sapros. Dr. Anupriya Kumar, she did her bachelor's in chemistry from Bates Pilani. Then she moved on to a doctorate degree in computational chemistry in South Korea on a full government scholarship during 2005 to 2009. After obtaining a doctorate, she worked at AstraZeneca as a computational chemist in antibacterial research during 2010 to 2013. Then to pursue drug-resistant bacterial research, she moved on to Nagawa University, Japan, initially as a postdoc and then as an assistant professor during 2013 to 2018. After gaining experience in computational drug discovery, she pivoted towards technical account management for scientific software as coordinator and now at OpenAI Cadence Design System in Germany. And Dr. Lucas Eberlin, he did his bachelor's and master degree in chemical biology at TU Dortmund University during 2010 to 2016, then move on to do a doctorate and postdoctoral in chemistry during 2016 to 2022 from the same university, funded by scholarship from the Resolve Cluster of Excellence. His main area of research is the development of statistical mechanical solvation models for quantum chemistry and their application to the investigation of protonation and conformal equilibrium of small molecules. Since 2022, he is an application scientist at OpenAI Cadence Design System Germany with expertise in conformal generation and molecular dynamic simulation for binding free energies. With this, we welcome you, Dr. Anupriya and Dr. Everman, and over to you both.
Thank you for the nice introductions, and thank you very much for the opportunity for inviting us. And today we'll be providing a very scientific talk starting from simulations, what is the application of simulation, and how can you detect cryptic pockets in novel proteins or already existing proteins for allosteric drug discovery. And today, because Lucas is an expert in molecular simulations, he'll be delivering the presentation. And this will be approximately for 1 hour. And last 30 minutes, please feel free to ask any questions to us. If we cannot answer today, we will reply to you in the email. So let's get started. I hope you enjoy the webinar.
Yes, thank you for having us. I hope you can see my screen.
Yes, sir. Yes.
And hear me. Does it work well?
Yes.
Thank you. Yeah, thank you again for the very nice introduction. I'm excited to be here and speak to you today, especially since it's already kind of late for most of you folks compared to us here in Germany. So thank you for staying and listening to us. The title of my talk is Drugging the Undruggable, because I'm going to introduce our method for detecting and ranking cryptic pockets to you, which we think is highly accurate. And I will present you some validation studies to underline this opinion. So to start with, here's the outline of my presentation today. I will start by introducing the KARAS system, an oncogenic protein long time considered undruggable to you, and show how cryptic pocket detection can speed up drug design drastically. We will discuss if and how we can use molecular dynamics simulations for cryptic pocket detection and focus on the weighted ensemble path sampling strategy, an enhanced sampling strategy that allows us to run our calculations more efficiently. This will lead us to our OpenAI solution for the protein sampling problem and subsequently to our method to detect cryptic pockets. We will have a larger scale validation study to show if and how well these methods work. And I would like to finish the talk by introducing our ligandability model to you. Such a model is important to be able to not only identify cryptic pockets, but also assess their ligandability to really find the pockets you can use for your structure-based design campaigns. Let's start with the KARAS system. Yeah, many of you likely know this system, so I'm just going to give a little bit of background in case you aren't familiar. KARAS was discovered in the 1960s, so the gene for it, and it acts as a regulatory switch by switching between the GTP-bound active and the GDP-bound inactive state. However, there are some oncogenic mutations, especially around glycine 12, that keep it in the on state for its entire lifetime. And this has many, many effects. Most notably, it causes tumors to grow. KARAS is one of the most commonly mutated oncogenes in solid tumors, with KARAS mutations being present in as many as like 20% of such tumors. The prevalence varies widely depending on the tumor type. For example, they are present in 85 to 90% of pancreatic ductal adenocarcinomas, so it really depends on the cancer type. And yeah, blocking the protein in the off state is a promising strategy for cancer treatment. I already said KARAS was discovered in the early 1960s via the gene, but it wasn't until the 1980s, early 80s, that it was found to be a very important target for cancer therapy. But it was considered an undruggable target for a very long time. It took around 30 years before people would find the switch 2 pocket here in this area of the protein in KARAS, which was discovered by work from Kieran Shokat's lab at UCSF in 2013, where he was doing some covalent fragment screening and found that this switch 2 pocket was potentially suitable for therapeutic design. Yeah, and just 10 years later, we now have 2 FDA-approved drugs on the market, sotorasib and adagrasib. And yeah, both of these bind to that newly discovered switch 2 site, and yeah, several other compounds are in the clinic. So if you think about the story of KARAS, the rate-limiting step for developing these therapies wasn't to find the actual drug-like molecule, it was to find the pocket. So what we would like to do at OpenEye is to remove this 30-year wait and to allow you to find this pocket basically with the push of a button. So there are a few questions that we are asking in order to answer the question whether or not we can develop a solution for detecting and ranking cryptic pockets. And these questions start out with, where might a ligand bind? Given a protein molecule, the simplest question is, on this protein, where are the possible sites where a ligand could potentially bind? And then you refine the question down to, where would a drug-like molecule typically bind? And this is really where you want to start a structure-based drug design campaign, using a pocket where you know a molecule would actually bind to. And finally, what happens if a molecule binds there? So some biological effect. And if we go ahead and start at the beginning, we have to have a crystal structure for any type of structure-based drug design. Usually it's an X-ray structure, but this can be really limiting. And we think that you can be trapped by the structure. And the reason why is that proteins actually move. And so I want to show you this quote from Galileo when he was held for heresy by the Catholic Church for supporting Copernicus's idea of heliocentricity, and yet it moves. So, and we need to take this statement to structure-based drug design. We can't be trapped by a crystal structure. These proteins move around, and we have to move them to see what they actually do. And you can do that by using molecular dynamics simulations. And it might take quite a lot of MD, or perhaps you have another way of doing it, and you can use slightly less MD and use something like a chemical probe. And yeah, we have ideas about how to do that. Now, once you've done some MD, you have to observe pockets, but that only tells you about where might a ligand bind. And now you have to actually rank the pockets to be able to tell someone that this is a really good pocket to start your structure-based design campaign. Sorry? Okay. So let's go ahead and try to answer the first question. How can we come up with a solution to the question, where might a ligand bind? In order to do that, we've been thinking hard about types of motion associated with cryptic pocket formation. Some of them are relatively simple, like sidechain motions, which happen in the order of nanoseconds, where you have a simple sidechain that need to move out in order for a pocket to open, or you have larger motions like loop motions, where you have an entire loop that needs to move out of the way in order for a pocket to open, or perhaps even the reverse process where a loop has to come back on top of a pocket to form basically a lid. Alternatively, there are longer timescale motions like secondary structure motions, where you have to move an entire secondary structure out of the way or bring it on top like a lid. And this is already a little bit more time consuming with microseconds to seconds, and outside of the scope of typical simulations. There are even more complicated scenarios where you have a secondary structure that needs to change, and you go from an ordered to a disordered state, or maybe the reverse process, or maybe even really long time scale motions like interdomain motions, where you have two parts of a protein that may be tethered or not, and they either have to come apart or move together to expose a pocket. And finally, there's another type of pocket that some people consider to not really be an actual cryptic pocket, but nevertheless, they are still pockets that need to be detected and ranked. And these are pre-existing pockets inside your protein. So we think about something like little holes inside the protein, like in Swiss cheese. And these are the types of motions that and pockets that we want to be able to detect. Before I show you how we actually do so, I want to talk a little bit about MD simulations, the timescales we can cover with them, and how it fits to the aforementioned timescales of cryptic pocket opening. So, what is an MD simulation? It's the simulation and analysis of the physical movements of atoms and molecules. We can observe the dynamic evolution of the system over time by applying Newton's laws of motion to the system at an atomic level. Most of the time, molecular mechanics force fields are used as the potential energy functions for MD simulations, but in principle, a variety of different potential energy functions, for example, derived from quantum mechanics, neural network models, or even hybrid approaches are possible. And what is really important, if we sample long enough, we can determine our system's macroscopic properties from such a MD simulation. Sorry. MD simulations are frequently used in drug design. We can use them to explore a huge variety of our system's properties with the prediction of cryptic pockets or pockets in general, and binding affinities probably being the main areas of application. But there are many more things for which MD can be an extremely helpful tool. Today, of course, we will focus on the detection of cryptic pockets. So let's go back to the timescales for even this cryptic pocket formation. While there are some motions like sidechain rearrangements, which are in the order of nanoseconds, most of these motions are in the microsecond to second range, with some going up to minutes. And here on the bottom, we have the wall clock time required to generate MD trajectories of a certain time with a typical desktop computer. We see that it takes a lot of compute and time to even simulate microseconds. Of course, we can speed things up by using proper high-performance computing infrastructure instead of desktop machines, but still, events that happen on the timescale of seconds to minutes can't be simulated with standard brute force MD. Even if it would be possible to run such a long MD simulation, it would not be guaranteed that the event we are looking for actually takes place, especially if the cryptic pocket opening is energetically penalized. To illustrate this, let's have a look at this alanine dipeptide example system. So here we have the free energy surface from multiple, many microseconds of biased MD, so replica exchange MD, and that's a lot of simulation needed to sample such a simple free energy surface, just the rotation around the phi and psi angles of this alanine dipeptide to identify this metastable state here. So we can imagine that for proteins, which consist of hundreds of amino acids, we definitely need methods to speed up the sampling process to be able to observe such slower motions. So again, sorry, here's the metastable state, and here we have the global minimum. One of the techniques we can use is coarse graining. Here, we have the time and length scale associated with motions important for drug design. We can again see that all atomic simulations will not help us to go to the timescales needed for some cryptic pockets to open. Coarse graining is a process of simplifying complex systems by representing them at a higher, more abstract level, focusing on large-scale behavior rather than the individual components. For example, by grouping several atoms of a chemical group into a single effective group. And this helps especially on the length scale, but slower protein motions are still hard to sample. So coarse graining can contribute to, but is not the full solution to properly sample the protein motions needed for cryptic pockets to open. We need to do smarter sampling as well. And there is a variety of enhanced sampling methods out there. It's important to carefully choose the appropriate method for each problem we want to solve using MD, and things like this decision tree can help us doing so. Some of these enhanced sampling methods use bias potential, as shown in the alanine dipeptide study, but we have decided to use a method that does not need to use such bias potentials and allows us to sample the protein conformational space efficiently. Before we get into the details of this method, I want to show you what we ideally want to see out of an MD simulation. Here we have the KARAS protein, and we will see the FDA-approved drug adagrasib. Let's have a look at the trajectory and focus on this region here on the right-hand side of the protein. We see a lot of conformational changes in the protein, including the opening of the switch to pocket and the drug binding event. So how are we able to simulate such a binding event? The method we apply at OpenEye is the weighted ensemble algorithm, which I'd like to briefly introduce to you. This is the free energy profile along an arbitrary progress coordinate x. We have two minima, and we can bin the free energy profile between those minima. We start with a simulation with a single stochastic walker, so an unbiased MD simulation, and run it for a very short time and check if it moved forward along our progress coordinate. Here it hasn't, and we split the trajectories to increase the chance of a simulation to reach the second bin. Our goal here is to always have two walkers in each of the bins. And it is important to keep track of the weights here. In this case, having two simulations starting from one with probability of 1 each of these two now have a probability of 0.5. After the next short simulation, one of our walkers reached the second bin, and again, we can split the trajectories to accelerate sampling. If a simulation moves backwards, as shown here, we can merge and split walkers again to always ensure we have 2 walkers in each of the bins. And over time, one of our simulations will reach the target state in bin 3. Again, it is really important to keep track of the weights, because this allows us to do a really cool thing. By properly keeping track of the weights, we can reconstruct equilibrium properties. So this method is able to accelerate the sampling along a progress coordinate without the use of a bias potential. And yeah, now coming back to our small example system, the alanine dipeptide, we can see that we only need to run 2 nanoseconds of weighted ensemble MD to properly sample the metastable state of the alanine dipeptide. To run the weighted ensemble MD, we use the VESPA package developed by Lillian Chong's lab at the University of Pittsburgh, which is now maintained by the Open Molecular Software Foundation. So this leads us to our solution for the protein sampling problem. In the alanine dipeptide example, we knew the progress coordinate needed to speed our calculation up. They were the phi and psi angles of the peptide backbone. But for protein sampling, we don't know these progress coordinates in advance. So the way we decided to do this at OpenEye is to use the intrinsic normal modes of the protein, as they will likely explore protein motions that are inherent to the molecular structure. The way we do this is we take our protein and we make a C-alpha based network model. From that, we calculate the Hessian matrix, we diagonalize it, and we get a set of normal modes which are expressed as the eigenvalues or eigenvectors lambda, which point in the direction of the motion you are interested in. And there is this really interesting relationship between the amplitude of a motion on the left-hand side, here the squared amplitude, and the energy of the motion, which is on the right-hand side. Also relating to the eigenvalue lambda and the frequency omega. And so the way you can think about this is that if you would fix the energy, then you get the largest amplitude from the lowest energy motion. Another way of thinking about that is that you fix the amplitude, and then the lowest frequency motion, the one that gives you that kind of motion that we think the Cryptic Pocket would open up, actually requires the least amount of energy. And that is what we are going to rely on, upon to open up our pockets. Normally, when people think about these normal modes, they talk about them in terms of the so-called fractional variance, which is shown here, and which is effectively related to the frequency of the motion. Fractional variance measures the percentage of the overall dynamics explained by a given mode. Usually they are shown sorted by the fractional variance, so the lowest modes have the largest fractional variance, which means they have the slowest, most large-scale motion, and they decay as the mode number decreases. However, what we've realized is that some of these low-frequency motions that give rise to the largest motion may not be appropriate for opening up pockets. So we use a different metric called collectivity, which measures how much of the protein is involved in the motion of a given mode. And if we use this collectivity and look how it lines up with the ranking based on the fractional variance, what you see is that some of the higher mode numbers actually have larger collectivity. Now, how does this actually look like? If we look at the eigenvectors projected back on the protein, the modes with the most collectivity, here the modes 14 and 15, shown here at the center, have eigenvectors that are effectively isotropically distributed across the protein surface. Whereas mode 12, which is really focusing on a particular region, doesn't have that, and so the collectivity drops. So what we are using for our exploration of protein motions is the most collective of the lowest frequency modes. How can we use this to actually get our protein to move around in a smart way? We are going to combine this weighted ensemble strategy, which requires a progress coordinate, and our normal modes, which will be our progress coordinates, and use that to explore the protein conformational space. We will start with an initial analysis to get our normal modes, so the eigenvalues and eigenvectors, and then we will run a short MD simulation for an ensemble of trajectories, and for each of these, we will recalculate the normal modes, which we originally calculated, and project those modes back on the original modes to see how far along a given progress coordinate we've actually moved. And then we cycle around this, and again, until we have done an appropriate amount of sampling. In this way, we are allowing the protein to explore conformational states in a really fast and efficient way. But of course, there's a catch. What if this isn't how you actually find a cryptic pocket? What we are doing here is to push the protein around and explore its conformational states, but maybe it requires some type of other effect to open up a pocket. In fact, it might be energetically unlikely to open up a pocket because it might need some kind of chemical probe. So what do I mean by this? For opening cryptic pockets, there are two axes that we'd like to think about. One is the conformational selection axis, where the protein is interacting with a probe molecule, which is water in a very high concentration. So you have a really greasy thing, the protein inside a really polar system, the water, so it moves around naturally based upon this. There is another axis, which is the induced fit mechanism axis, which requires a chemical probe to be there in order for the energetics to be more likely. And it is possible that the cryptic pocket opening happens somehow as a linear combination between these two. So what we are going to do for this induced fit mechanism, what goes here on the x-axis to be our molecular probe. It could be something as simple as xenon or two perhaps open pockets. That's the question we have. Is it helpful to open pockets? We think, based on our knowledge about this specific atom, that it is. There are really these two ways to open a cryptic pocket, as just explained on the last slide, but I'd like to go into a bit more details. One, we have a conformational selection mechanism. This means that the protein opens up by interacting with water. What you are seeing here are residues shown in orange that change their solvent exposure based on the interaction of the protein with water itself. The induced fit mechanism is slightly different. Here you see orange residues that appear not because they are solvent exposed, but rather because they are binding probe molecules. These can be experimentally verified, both of these mechanisms, and they have an observable, which is the ligand concentration. So if you're thinking about the conformational selection mechanism, there is no ligand necessary to be present, so you have no ligand dependence. However, if you have an induced fit mechanism, there's a strong ligand dependence. If you add more ligands to it, it is more likely to find a suitable protein nearby, and therefore you have a strong ligand dependence on the pocket opening. That's the way we see these two mechanisms working. Let's go back to the idea, can we really use xenon as a reasonable chemical probe to induce cryptic pocket opening? We think we can, and that's based on some observations. So number one, we know that xenon acts as a general anesthetic. So what does this mean on a molecular level? If you have an ion channel like this GABA A receptor and you add xenon to it, it will cause the receptor to close, and you can see the macroscopic change happen in the current that goes across the membrane. And so it stops the flow of ions, and that affects the central nervous system. Another aspect is that we think is that xenon acts as a neural protectant. Xenon promotes neurogenesis and it prevents apoptosis. Because of that, we know that it has some interactions with proteins that cause a dramatic shift in their conformational equilibria. And because of these two findings taken together, we think that xenon could potentially stimulate pocket opening through an induced fit mechanism. Sorry. With these, we come to the methods which we at OpenEye use for detecting our cryptic pockets. We will use them after we applied our protein sampling method. The first method is called exposon analysis. This was developed originally in the lab of Greg Bowman, and it uses correlated changes in solvent exposure, as I explained before, as the way to define what residues are involved in cryptic pocket opening. And we were highly inspired by this method and developed our own method, which is called dynamic probe binding analysis, where you can run a simulation in the presence of xenon using our protein sampling protocol. And we have an analysis method that can look at the correlated changing in the binding propensities of xenon to the protein. And these are two alternate methods. One, the exposon analysis that allows us to look at the conformational selection axis, and the dynamic probe binding analysis that allows us to look at the induced fit mechanism axis. I want to show you how our method works by using a case study on KARAS, which we quite recently published. As a reminder, KARAS acts as a regulatory switch by switching between the GDP-bound active and the GDP-bound inactive state, and locking the protein in the off state is a promising strategy for cancer treatment. On the left-hand side, we see the KARAS APO structure, so without a ligand, and the holo structure with a ligand bound on the right-hand side. And the main difference that we see is that there had to be some dynamics for this pocket to open, and the time scale is potentially quite long, up to seconds, which is far beyond what typical brute force simulations can do. And also the length scale is quite long. You have to move loops around, some intra-domain motion and other things. So how we are able to do this? We will use our normal mode-driven weighted ensemble method to hopefully open up these pockets, and then we are going to take our previously explained methods, the exposon and dynamic probe binding analysis, to analyze the protein and see if we are able to detect these pockets. And this is the result of this study. The switch 2 pocket is opened in both cases, so we quite nicely see the residues involved in the pocket opening are highlighted by both of these methods, and they really quite nicely open the pocket where this Mirati compound binds. We also see that this buried tyrosine 71, which is more pointing inside the protein in the apo structure, is solvent exposed in the holo structure, so after the simulation. So this tyrosine 71 may act as a gatekeeper for the cryptic pocket to open. From our xenon binding analysis, we actually found 8 interesting sites. So it's important for us to figure out if and which of them may be false positives. And before we had developed our ligandability model, we were afraid to give you too many of these false positives, and we used this probe occupancy analysis to rank pockets. we can calculate an energy for the xenon binding by comparing the enrichment of xenon at the potential binding sites to the bulk occupancy. Let's see how many of these binding sites are experimentally validated, and start with the binding site 1. So the top ranked pocket from this dynamic probe binding analysis is actually the binding site where the 2 FDA-approved drugs bind. And we even see that the xenon binds in exactly the same region where this apolar part of the inhibitors binds. The binding sites 1 to 3 combined form the binding site which is used by this peptide inhibitor. I think one of the larger US pharma companies has worked on that. And we also find ranked 4th a binding site which is the switch 1-2 pocket. It's exactly on the opposite side of the switch 2 pocket, and this is a more shallow pocket where an analog compound of the actual FDA-approved drug can bind. So we think the results of this KARAS case study are great, but of course we wanted to do some more larger scale testing and not rely on the KARAS alone. So we prepared this validation data set that includes lots of different types of proteins with different types of pockets, covering the motions mentioned at the beginning of the talk. Pockets that open up by sidechain motions, loop motions, secondary structure motions, secondary structure changes, interdomain motions, and even some deeply buried pockets. We want to use this data set to answer the first question: where might ligands bind? And we see that we are able to identify 91% of the pockets that we tried. The failure cases here and here include one secondary structure change where we need to go from an ordered state over a disordered state to another ordered state. Naively, we would like to blame the force field for this, since force fields are typically parameterized in a way that they keep secondary structures intact, but we can't necessarily do this, so we need to improve our method here. The other failure is a deep pocket, where we are not able to find a pre-existing deeply buried pocket in the protein. Maybe we need to increase the concentration of xenon to be able to properly detect it. Both of these fail cases are from the same protein, so we are still investigating how we can improve here. Overall, we were quite happy with the results of our validation study, but we need to do more work to answer the next question: where does an actual drug-like molecule bind? Not where just where might it bind, rather where do I start my structure-based drug design campaign? We need a method that allows us to separate good from bad pockets. We have been developing a model that gives you a sense that the pocket is more than just finding a groove on the surface of a protein. Before I go into the details of our model, I want to give you a very short overview about ligandability assessment methods, just to give you a better idea what our model does. A lot of work in the field comes from the recent years. It basically started around 2007 with the PLB index model, where they trained a model on just 41 structures and asked the question: what is the propensity of a given amino acid to be inside a binding pocket? So they look at the pocket and they can say, for example, proline is very unlikely to be there, tryptophan is really unlikely to be there. And based on this residue composition, I can give this pocket a particular score. About 10 years later, they developed P2Rank. they upped the training set by around half an order of magnitude to 251 structures. They used a random forest model, and instead of the surface residues, they looked at surface points. Each point on the surface of a pocket gets a certain score. And you rank the pocket based on the sum of the scores of the surface points. And then we have this really cool and new model that came out just last year from the Tivari lab called GRASP. And this is a graph-based neural network model which uses surface atoms and was trained on a way larger data set of nearly 17,000 structures and 26,000 binding sites. Yeah, it's a nice model, but we would like to argue that there are still some issues with this, not because the model is bad, but we think that the training data were not necessarily appropriate to make a ligandability model. The question we have is, do the existing models use truly non-redundant training sets? And how they split their data sets was basically based on two criteria. One was the protein sequence identity. The other one was the structural similarity to a given protein family. These have some issues with them, and I'd like to point out why we think that's the case. Point one, we don't think protein sequence identity itself is sufficient because it doesn't reflect the protein's 3D structural similarity. What do I mean by this? I mean, the same sequence can adopt two structurally completely different conformations. A classic example here is calmodulin, which has a totally different structure if it has a calcium bound and when it doesn't. And also you can have different sequences that can lead to the same fold, which can lead to the same enzymatic activity. And there is another issue if you look at protein families, because many protein families have diverse binding sites, but they bind to the same types of molecules. So you might be overfitting on some of the same chemistry, and we think that this is what happens in some of the models that are out there today. So if we look at the SCPDB bind dataset, the dataset on which GRASP is trained, it is unfortunate, but around 50% of the training data comes from only 3% of the ligands. So there was not enough attention paid to the ligand chemistry when they did their splitting. And yeah, what kind of ligands are those? Well, they are mostly nucleotides or nucleotide derivatives. And we think this is a problem when you try to assess pockets that not necessarily bind nucleotides. We think a good example is our favorite KARAS system. If we apply the GRASP model to the system to see which of the three pockets that we know about is the most ligandable, it will clearly recognize the GDP binding site as the most ligandable. But it will ignore the switch 2 pocket, which is clearly an FDA-approved drug binding site, as well as the switch 1-2 pocket. And this is an issue if you want to use a model like this for prospective work. How do you actually identify binding sites that are not nucleotide binding sites? To answer that, we created our own data set, which we think removes some of these issues. We used pocket similarity for preparing our data set and used splitting based on that to avoid leakage from our test to our training set. We rely on a method developed many years ago at OpenEye, which is called SiteHopper. In SiteHopper, we use the shape and the electrostatics encoded as the so-called color features, which are chemical features, and use this to create protein pocket patches, and then afterwards we do an overlay of the pocket patches to measure pocket similarity. And For our data set, we deduplicated our pockets based on this pocket similarity, which is not necessarily protein similarity, sequence similarity, or protein family similarity. This way, we curated a non-redundant protein ligand dataset consisting of approximately 1900 structures. We used this dataset to train our model on. We did the split of to approximately have 1500 protein ligand complexes in the training data and 400 in our test dataset. This is what it looks like. We have a protein pocket surface patch representation. And here, this is the positive control. We use a protein ligand system and consider the surface area within a certain distance criterion around the ligand to be a positive protein patch. And we have a negative control patch, not going into too much details here, but this is a part of the exactly same protein, which has a similar curvature and size as the positive protein patch. And this is what we are calling our negative control. We can take this and we decorate the surface based on some distance criteria. And we have a feature vector, which is based on some point features that are taken from our toolkits and are inspired by the P2Rank model. And we also were inspired by GRASP and used some atomic features, which we are projecting back on the surface, again, using our toolkits. So these toolkits are one of our products. There are programming interfaces that allow you to basically do anything we can imagine with molecules in a computational way. For this work, we mainly used our tool QuackPack, which is made to assign charges, predict protonation states and tautomers, and calculate pKa values. From here, we take the point feature electrostatic potential and the partial charges, which are an atomic feature. And the other toolkits are Spicoli, which is used to generate the surfaces and construct grids, and ZAP, which is used for most of the electrostatics work. It's a Poisson-Boltzmann solver that can calculate a variety of descriptors, including solvation and transfer energies, and is here mainly used to identify solvent-exposed atoms and calculate their density. And then we go ahead, take all of our positive and negative control patches, we featureize them, and we train a model, either a random forest model or a Gaussian progress regression model. We found that random forest models tend to be a little bit more performant. So all the data you will see here will be from such a random forest model, but in general, both methods work quite well. This is how the model looks like. We have a set of surface points similar to P2Rank, where every surface point has its little bit of pocket score, and we take the sum of the squares of these points and call this our total score, and then we normalize it by the number of surface points so that we can compare pockets of different sizes. We don't want score to grow just because the size of the pocket grows. Rather, we want to define what is a ligandable pocket regardless of the pocket's size. This is how the data looks like. In gray, you see the negative control patches, and in red, the known pockets from our data set. And we see that there is a separation between them at around 0.3 in our normalized ligandability score here. Also, our AUC, so the area under the receptor operator curve, which basically measures how well you distinguish true positives from false positives, is really high with around 0.97. So we think this model is really good in separating our pocket patches from the negative controls. In order to find an actual breaking point, we use the so-called base factor, which is the ratio of two likelihoods of probabilities. The idea is that if you have the same data given a model which is liganable or not liganable, how much evidence do you have that the model is predictive or not? And a Bayes factor higher than 1 gives you evidence that your model is really predictive. And we use a normalized ligandability score of 0.72 as a cutoff to consider a pocket ligandable, which corresponds to a Bayes factor of 3.2. Okay, now let's look at under the hood of our ligandability model. And I would like to remind you of this useful quote from George Box that says, all models are wrong, some models are useful. So we ask the questions, are we biased towards any kind of pocket properties? First thing we investigated is we plotted the pocket similarity with the training set on the y-axis and the predicted pocket score on the x-axis. We see that for the known pockets, there is no dependence on the similarity with our training set. So this is cool. And the next properties we are looking at are the buriedness and the pocket polarity. So the buriedness is effectively the difference in solvent accessible surface area in the bound and unbound state. And here we see that most of the high scoring protein binding sites tend to be buried. And this is actually a reflection of the population in our training set. So also most of the known pockets in our training set are more buried. The next thing is pocket polarity. And for pocket polarity, we don't observe such a trend. So most of the known pockets have kind of a similar polarity, and this is also very well reflecting the population in our training data set. So our ligandability model is biased towards buried pockets, which can be good, can be bad. And so we wanted to see how well does the model perform on real binding sites. So promising sites like the FDA-approved drug binding pockets. And here we see that 81% of the 121 drug binding pockets are predicted to be ligandable. But we see that we also have some false negatives down here in this score region. So these false negatives are binding pockets that bind either due to stabilization of a cofactor, contain macrocyclic drugs, peptides or peptidomimetic drugs, covalent drugs, or some other things. So most of these data we have purposely removed from our training set because we don't have enough data for it. So we can improve our model for these types of drugs if we have more training data, but we are not surprised that we have some false negatives here. And now what I've just shown you is the evaluation of our ligandability model on crystal structures. And it works great on crystal structures, but crystal structures are showing very nice pockets which are perfectly stabilized by the cognate ligand. So the next question is to be able to do prospective work. How well does the ligandability model work with imperfect pockets, which we find from our protein sampling workflow, which we previously discussed? And so we tested the model on our known cryptic pockets from the validation study. And we see that most of these pockets are above our score threshold and should be ligandable. The only false negatives that we have are the same type of false negatives we had with the FDA-approved drug binding sites, peptides or peptidomimetics, which we know are missing in our training set. So we are not surprised to see that we miss them here again. If we go back to the KARAS system, and if you remember, the GRASP model was only able to identify the GDP binding site, then we see that our ligandability model ranks the FDA-approved drug binding site, the switch 2 pocket, on top, followed by the more shallow switch 1,2 binding site. So we know our ligandability model has some biases and imperfections. Shallow pockets will likely get a lower score. It's slightly biased towards buried pockets, and the model will likely fail for certain binding sites, which are ligands that are stabilized by an additional cofactor, peptidomimetic and macrocyclic drugs, or covalent inhibitor binding sites. So some of them, just missing a feature representation in our model, and others will we miss because of insufficient examples in the training set. So back to our questions, where might ligands bind? This led us to the development of a protein conformational space sampling workflow, which I hope I could show you works very well for most proteins. The second question was, where does a drug-like molecule bind? This led us to the development of our ligandability model. And the third question is, what difference would a ligand make if it does bind? And that's a larger problem of biological efficacy. And we are always looking for partners that help us to answer such questions. So if you're willing to work on something like this, please get in touch with us. So to summarize, we developed an excellent extended dynamics sampling protocol for the detection of cryptic pockets. We use xenon as a probe for the detection of cryptic pockets in the induced fit mechanism. We are writing a manuscript at the moment, which will hopefully be published soon. And we developed a ligandability model, addressing a general Achilles heel of pocket finding to rank the pockets for ligandability. With this, I am at the end of my talk, and I want to thank the people at OpenEye who actually did the work. So this is mostly the enhanced sampling group led by David Labart. This project was a multi-year collaboration with Black Diamond Therapeutics, and also inspired by some discussions with David Lawson from Mirati Therapeutics. We'd like to thank the Westpark community. Without their work, our work would not have been possible. And a really cool thing is all of this work is built into pre-existing workflows on our molecular design platform, Orion. So everyone can get access to it to make your drug design campaign